As we age, our DNA is exposed to damage from both external and internal sources. DNA repair is imperfect, resulting in mutations like double strand breaks and translocations. This leads to epigenomic drift, where the epigenome changes over time. Research suggests that there is a correlation between epigenetic changes and aging-related diseases. For example, certain age-related diseases like cancer, cardiovascular disease, and neurodegenerative disorders have been associated with specific epigenetic alterations. Epigenetic drift is thought to contribute to the aging process. Enter Dr. Vera Gorbunova, a professor of biology at the University of Rochester and co-director of the Rochester Aging Research Center.
My name is Vera Gorbunova. I'm professor of biology and medicine at the University of Rochester in Rochester, New York. I've been studying aging and longevity for more than 20 years, and the goal of my research is to develop interventions to extend human health span and lifespan. One of the areas that we study are proteins called Sirtuins that many years ago, the group of Lenny Guarente with David Sinclair discovered that adding extra copies of this gene or boosting its activity extends yeast lifespan. But this is in yeast, that's a model organism. What about humans? In humans, we have 7 Sirtuins, and of those 7, the one that seems to be most relevant to the process of aging is named Sirtuin-6. And my team is studying Sirtuin-6. What we showed is that activity of Sirtuin-6 correlates very strongly with maximum lifespan of animal species, and animals that are long-lived have much stronger Sirtuin-6. Also, adding extra copies of Sirtuin-6 in transgenic mice extends their lifespan. So this is quite amazing. What does Sirtuin-6 do? It has many beneficial functions. It improves DNA repair, so it helps us avoid mutations and genomic instability. It also maintains proper chromatin organization in the cell so that correct genes are expressed, and then genes that should be expressed are silenced. And very importantly, Sirtuin-6 silences line 1 transposons, these genomic parasites that, again, my team has shown to cause inflammation, and boosting Sirtuin-6 can reduce inflammation. So how do we increase Sirtuin-6 activity in humans? There are many researchers looking for activators of chemical activators of Sirtuin-6, including my group. But of course, these chemical activators have to be very safe. And there are already some natural compounds that have been identified that can stimulate Sirtuin-6 activity. So we've been screening different preparations of seaweed and fucoidan, and we identified those that are really strong inducers of Sirtuin-6 activity. A molecule from seaweed that is very safe and very beneficial for human health. I am a proud member of Do Not Age Advisory Board because this is a company that brings these anti-aging interventions to people.
Dr. Gorbunova collaborated with a supplement company, Do Not Age, to test and provide a product derived from select batches of brown seaweed that have been verified to activate Sirt6. To ensure efficacy in activating Sirt6, Gorbunova's lab subjects every batch of seaweed to rigorous testing, selecting only those that activate Sirt6. A double-blind study was done with mice. Those given the Sirt6 Activator showed reduced frailty with age and significantly extended lifespans. The promising outcome from this study prompted the start of a clinical trial on elderly patients who have undergone chemotherapy aimed at evaluating the effects of Sirt6 Activator. With this exciting development in longevity, I've personally started taking the supplement from donotage.org called the Sirt6 Activator. Fucoidan, specifically from the variety of brown seaweed Fucus vesiculosus, is approved for consumption by the United States Food and Drug Administration, FDA, and is generally recognized as safe. I will track and monitor a number of biometrics, including blood pressure, weight, BMI, sleep quality, resting heart rate, body fat, and exercise endurance. To try and keep other factors from affecting the results, I will not change my diet or my exercise habits. I'm especially interested in how my endurance may be affected. One of the studies on Sirt6 showed increased VT2, the level of exertion at which lactate starts to build up, and a person needs to breathe heavily. I'll report my results after 1 month, 3 months, 6 months, and 1 year. So stay tuned for more, and as always, thanks for watching.